Hello everyone, welcome back to the session. So in the last class we have discussed about the cell theory, okay, and some of the MCQ. So in today's class we are going to discuss about the prokaryotic and eukaryotic cell organization. Not only prokaryotic and eukaryotic cell, but also we are going to discuss about what is mesokaryotic cell. Okay, without any delay, we'll start it. So first, prokaryotic cell organization. So prokaryotes are the organisms with a primitive nuclear organization. The word pro refers to primitive. The word pro refers to primitive, and the word karyon means nucleus. Okay, so those organisms which are having a primitive nucleus, we are classified that organisms under the prokaryotic cell organization. Okay, so based on the nuclear organization, we have categorized the organisms into prokaryotic, mesokaryotic and eukaryotic organisms. So here the true nucleus will be absent and the nuclear membrane is also not present. Nucleus is absent and the nuclear membrane is also not present in case of prokaryotic organisms. And what if with respect to this genetic material? So the genetic material is present in the region. So the region where the genetic material is present is said to be as nucleoid. Okay, so here the nucleus is absent, the proper nucleus is absent. So basically the genetic material are present in the nucleus, but in prokaryotic organism, as the true nucleus are absent, the place where the genetic material is present, that particular region is called as a nucleoid. Okay, so here the nucleoid is not a membrane bound and hence it is not a true nucleus. Okay, so the nucleide is not a membrane bound, hence it is not a true nucleus. So the DNA is single here. So the DNA in human beings, it will be a double stranded DNA, but here it will be single, and basically the DNA will be linear in structure, but in prokaryotic organism it will be circular, and also it is naked in the sense, so it is not associated with any histone proteins. That is what naked. So basically the DNA in human beings, it will be attached with the histone proteins. So histone is the one wrapping the DNA. Okay, but here in prokaryotic organism, the DNA is not packed with any of the histone proteins. The DNA is a negative charge and the histone will be positive charge. So these two charges attract with each other and they are wrapped within a nucleus. But in prokaryotics, it is not packed with any of the histone proteins. So that's why we are saying this DNA in prokaryotic organisms will be naked. Now, so this here the cell membrane is also absent, true nucleus is also absent, the membrane bound organelles is also absent in case of prokaryotes. And the prokaryotic cells are structurally simple but smaller in size, we know that. And what are all the organisms which are placed under the prokaryotes? Bacteria, cyanobacteria. So cyanobacteria is called as the blue-green algae, mycoplasma, rickettsia and spirochetae. So these are all the organisms, we can classify them under the prokaryotes. Now mesokaryotes. So mesokaryotes was proposed by Dodge and the co-workers. Okay, but mainly Dodge. Okay, so prokaryotes means a poorly developed nucleus. Eukaryotes in the sense a well-developed nucleus. So the nucleus which is present in the mesokaryotes, which is not well developed, which is not poorly developed. Okay, so it is evolving. So this is an intermediate characters between the prokaryotes and eukaryotes. So these are all the organisms. We can't say that as a true prokaryote and true eukaryote. So that's why we can classify them under the mesokaryotes. So these possess a well organized nucleus. So they are having a nucleus with a proper structure. Also they are packed with the nuclear membrane. And here the new DNA is organized into the chromosomes. The chromosomes will be they are having a DNA, and but histone proteins are absent here. See here the DNA is organized into a chromosomes, but it is not packed with the histone proteins. So that is why they are having a chromosomes, that this is not considered as a prokaryotes, and they are not having a histone proteins. We can't say that as a eukaryote. So that's why we can call them as a mesokaryotes. Okay, so here the cell division occurs by amitosis. Basically the cell division we can categorize them into mitosis and meiosis. Here the mesokaryotes, the cell division occurs here by the amitosis process, similar to the prokaryotes. Okay, so prokaryotes also reproduce cell divide. The cell will divide in the prokaryotes in the process with the process of amitosis. Like in the same way, mesokaryotes is also dividing with the process of amitosis. So this mesokaryotes bridge the gap between the prokaryotic and the eukaryotic organization. So what are all the organisms which is placed under the mesokaryotes? So protozoans, that is the protozoan category, specifically Noctiluca. So this is the organism which is coming under the protozoan, and this protozoan is coming under, it's called them as the mesokaryotes. And phytoplankton. So what are all the phytoplanktons? Gymnodinium, Peridinium. So these are all the examples. And dinoflagellates. Again, this will be the protozoans which is coming under the mesokaryotes. Now comes the eukaryotic cellular organization. So this eukaryotes or the organisms possess the true nucleus. Okay. So this is the true nucleus. So the word "eu" means true. Karyon means nucleus. So this is the true nucleus, and here the nucleus is surrounded by a membrane, that is a nuclear membrane, and here the DNA is organized into a chromosomes, and it will be completely packed up with the histone protein. So that is why we are calling them as the eukaryotic organization, eukaryotic cell organization. See, in case of prokaryotes, they didn't have any of the the DNA is not organized into a chromosomes, and the DNA is not associated with the histone proteins. In case of mesokaryotes, the DNA will not be associated with the histone proteins, but it will be organized into a chromosomes. But in eukaryotes, the DNA will be associated with the histone proteins, and it will be organized into a chromosome. So this is a proper structure. We can call them as a nucleus. Okay, proper structure of nucleus. And also we are having a proper membrane bound organelles. We are having a membrane bound organelles in their cytoplasm, showing their structural complexity. As all these different cell organelles are present with the nucleus, nucleus membrane, cell and cell membrane, we are saying this eukaryotic cell organism is little bit complex when compared to pro and mesokaryote. Okay. Now, some organelles are believed to have originated by endosymbiosis, which means you should know that some of the organelles which is present in the cell, eukaryotic cell, are originated by an interaction, and that interaction is called as an endosymbiosis interaction. What is meant by endosymbiosis? Symbiosis means one organism will be interact with the other organism. Okay, so that may be a mutual. That may be one organism will give something and take something with the associated organism. So that is what a symbiosis. Endo means within the organism. So that is what an endosymbiosis, a cell living inside the another cell. So that is what a cell organelles. The organization of cell organelles will be under the theory which is called as an endosymbiotic theory. So those organelles, so what is the very important organelles which is support for the endosymbiotic theory? Mitochondria and the chloroplast. This mitochondria and the chloroplast strongly support the endosymbiotic theory. So the eukaryotic cells show a high level of organization and compartmentalization. Okay, so the eukaryotic cells shows high level of organization, which means they are having a cell, and this cell is covered by a membrane which is called a cell membrane or a plasma membrane. In the middle they are having a nucleus, and this nucleus will also be surrounded by a nuclear membrane, and between the nucleus and the cell membrane we are having a cytoplasm. In the cytoplasm many organelles are floating in it. Okay, so the most importantly the mitochondria and the chloroplast, they are forming a how these organelles have been evolved. So those the theory which explains the origin of the mitochondria and the chloroplast will be the endosymbiotic theory. Now, so what is this endosymbiotic theory? So two eukaryotic organelles, that is nothing but a mitochondria and the chloroplast, believed to be the descendants of the endosymbiotic prokaryotes. So some of the prokaryotes, Okay, so prokaryotes which is developed within the eukaryotes. Okay, so the eukaryotic organism. So inside the eukaryotic organism, some prokaryote will live. Okay, that's what endosymbiosis. So this prokaryotes is the one we are evolved into mitochondria and the chloroplast. Okay, so that is what we are saying. Two eukaryotic organelles, that is nothing but a mitochondria and the chloroplast. They believed to be the descendants of this endosymbiotic prokaryotes, which means what? The ancestor of mitochondria and the chloroplast will be the prokaryotes. This is what the endosymbiotic theory. Now, the ancestors of eukaryotic cell engulf a bacterium and the bacteria continue to function inside the cell. Later, this bacteria evolved into a cell organelle like mitochondria and the chloroplast. This is what the endosymbiotic theory. Now we are going to excuse me. Now we are going to compare the difference between this prokaryotes, mesokaryotes and eukaryotes. Okay. So what will be the cell size? So the prokaryotes, the cell size will be approximately 1 to 5, and the mesokaryotes will be the approximately 5 to 10, and the eukaryotes will be approximately 10 to 100. And then what will be the nuclear character in case of prokaryotes, mesokaryotes and eukaryotes? So here the nuclear character of prokaryotes will be the nucleoid, which means there will be no proper nucleus and there will be no proper nuclear membrane, but instead the area where the genetic material is present, that area is called as a nucleoid. Okay, so the mesokaryotes means they are having a nucleus and they are having a nuclear membrane but without a histone protein. And in the eukaryotes, they are also having a true nucleus with a proper nuclear membrane. Okay, and in the DNA will be circular in prokaryotes and linear in mesokaryotes and the eukaryotes. And in the prokaryotes, it is usually circular and there will be no histone proteins present in case of prokaryotes. But in mesokaryotes, the DNA will be linear without histone proteins. In the eukaryotes, there is a linear DNA but with the presence of this histone proteins. Okay, so you should know what will be the charge of a DNA, that is negative, and the proteins, the charge of the histone protein will be positive. Together they attract with each other with the help of the charge. The histone proteins will wrapping up the DNA and it is organized into a chromosomes. Okay, this is what a function of histones. Now what is with respect to this RNA and protein synthesis? So in case of prokaryotes, the transcription and translation coupled into cytoplasm. So basically we are having a replication process, transcription and translation process. Okay. So in the replication, DNA is converting into a DNA. Transcription, it will be the DNA will be converted into an RNA, and in the translation the RNA will be converting into a protein. Okay. So in the transcription, so in prokaryotes, the replication process, the transcription process and the translation process is a simultaneous process, and we can say that as a coupled reaction. In case of mesokaryotes and the eukaryotes, the replication has to be completed. The DNA has to be converted into one DNA, and from the DNA, the RNA strand has to be synthesized. That process is called as a transcription. Only after the transcription process has been completed, the translation process is going to start. Okay, but in case of prokaryotes, the transcription and translation will work simultaneously. It is a simultaneous reaction, but it is not in case of mesokaryotes and eukaryotes. Okay, now the ribosomes in prokaryotes, mesokaryotes and eukaryotes. 70 is in prokaryotes and 80 is in mesokaryotes and the eukaryotes. Okay, so we have separated this 50 s plus 30 s. You should not say that as an 80 s. Okay, so this S is a sedimentation rate. Okay, so this is a larger unit and this is a smaller unit. As this sedimentation rate, if we minus the sedimentation rate, the remaining will be the 70 s. So this S is considered as a Swedberg unit. The scientist name is Swedberg unit, or we can say this that as a sedimentation rate. Okay, so that is what 80 is. 60 plus 40 is 90. We can't say this as a 90 years, so that is a sedimentation rate, so we can say that as an 80 years. Same with respect to the eukaryotes. So what is mean by this membrane bound organelles? The membrane bound organelles will be absent, completely absent in case of prokaryotes, and it is present in eukaryotes, but it is not well developed when compared to the eukaryotes. So in eukaryotes we are having a numerous membrane bound organelles. What will be, how this prokaryote will move, how the mesokaryote and the eukaryote will move. The flagella is the flagella is the locomotory organelle in case of prokaryotes. In meso and eukaryotes, flagella as well as the cilia will be considered as a locomotory organelle. And what will be the level of organization? So basically we are having four different types of cellular organelle, but the majorly two. Okay, one is unicellular and multicellular. In case of prokaryotes, it will be usually unicellular organization, which means it is made up of only one cell, and that one cell is responsible for doing all the physiological functions. In case of mesokaryotes and the eukaryotes, they are unicellular. Both are unicellular as well as multicellular, but this mesokaryotes will be unicellular and it is colonial. Colonial in the sense we are having two important words. One is solitary. Solitary means individual, okay, and colonial means it will be live in a group, but this mesokaryotes will live in a group. This eukaryotes, it may be unicellular or multicellular and it will be colonial in nature. Now cell division. This prokaryotes will divide by this binary fission, which means simply it is a cell. This cell will be elongate its nucleus. Elongates its nucleus, and there will be in one cell there is two nucleus, and after that this cell will be divided into two different cells. So this is what a binary fission and this is what a prokaryotes in case of. So now in mesokaryotes, they are divided by amitosis or binary fission. In case of eukaryotes, they will be divided by the mitosis and the meiosis. And what is the example which is coming under the prokaryotes, eukaryotes and the mesokaryotes? Bacteria, that is archaebacteria and eubacteria will be coming under the prokaryotes. In mesokaryotes, the dinoflagellates and protozoa is coming under the mesokaryotes, and the fungi, plants and animals, these kingdom are coming under the eukaryotic organisms. So that's all about the basic difference between prokaryotic cell, eukaryotic cell and mesokaryotic cell. Now another important difference between what is meant by plant cell and what is the animal cell. Okay, so usually when compared to the animal cell, plant cells are very larger in size. Okay, and also the cell wall is absent in the animal cell, but instead of cell wall there is a cell membrane is present in case of animal cell, but in the plant cell the cell wall is present and the cell wall is made up of the cellulose. Okay, so the cellulose, that is hemicellulose. Okay, by cross linking with each other, the cell wall is present in case of plant cell, but it is absent in the animal cell. Outside the plasma, they are having a cell wall, cell membrane and the cell, but we are having only the cell membrane, not a cell wall. Okay, so and this consists of middle lamellae, primary cell wall and the secondary cell wall together. That is why we can see that trees are very strong, their cell wall is very strong. That why? Because it is made up of a cellulose and the lamellae. Next, another important thing will be the plasmodesmata. Plasmodesmata, it is nothing but a gap junction. Okay, gap junction means the cytoplasm of one cell will be connected with the cytoplasm of another cell. In case of plant cell, we are calling them as a plasmodesmata. This is a specific structure which is present only in the plants, but it is absent in case of animals. And the chloroplast is present and it is absent in the animal cells. Why? Because as the chloroplast is present, photosynthesis process will be done by the plants, but as it is absent in animals, we can't generate our own food materials within our body, but instead we are dependent on plants and other organisms. Okay, vacuole is very large and it is permanent in the plant cell, but here it will be very small and temporary. Okay, now tonoplast. Tonoplast means the wall of the vacuole is considered as a tonoplast. We are not having a vacuole, so obviously tonoplast will be absent in animal cell and it is present in the plant cell. And the centrioles is absent in plant cell but it is present in an animal cell. Why? Because the centrioles will be present only when this is very important with respect to the cell division. This centrioles will be present only in the motile cells of some lower plants, okay, but it is not present in all plants, okay, but usually it will be absent. Centrioles are absent in the plant cells, and the nucleus usually pushed to the periphery due to the large vacuole. So consider this as a plant cell, this is a vacuole. As the vacuole is occupies a major portion in the plant cells, the nucleus will be in the peripheral region, okay, but here the nucleus in the animal cell will be in the central region. Now, the lysosomes are rare in case of plant cell and it will be very well developed in the animal cell. So, the lysosomes are considered as a suicidal blocks. As its importance is very important in case of animal cell, it is present in the animal cell and it is absent in the plant cell. And there is a storage material, and here the storage material will be the starch grains, but here the storage material will be the glycogen. The excess glucose will be converted into the glycogen, and this hormone involving here will be the insulin, and this glycogen will be stored in the liver and the muscle in case of animal cell. But here the storage material will be the starch, right? So that's all the difference between the plant cell and animal cell. Another important area where we can expect more question will be the difference between archaebacteria and the eubacteria. What will be the difference? So archaebacteria and the eubacteria in the sense, so these are all the bacteria, these are all not an usual bacteria, it is an unusual bacteria. The eubacteria will be, U means true, this is considered as a true bacteria. So what is the difference between these two? Okay, so the domain, it will be archaea and this domain will be the bacteria. So the specific feature, the special feature that the archaebacteria is possessed is, so they can live in an extreme condition. Extreme condition means they can live in extreme hot condition, they can live in extreme cold condition, they can live in high pressure condition, and they can live in a low pressure condition. They can also have a capacity where the salt concentration is very higher, and they can also have the capacity to live where the methane is higher. Okay, so they can live in an extreme habitats. But these are the bacteria which live in a normal environment, we can call them as an eubacteria. Okay. So that is what extreme habitats means: hot springs, salt lakes, anaerobic areas. So to be an add point, they can also have the capacity to live in an area where there will be no oxygen. Okay, but in eubacteria, they can live in a normal environment, they can live in a normal environmental conditions. So that's why we are calling them as a eubacteria. Okay, so the cell wall, the archaebacteria, their cell wall, they are not having peptidoglycan. Okay, so the peptidoglycan, this is a NAM and the NAG. NAM plus NAG, okay, N-acetyl glutamine, N-acetyl muramic acid, okay. So these are all the components together, we can call them as the peptidoglycan, NAM and NAG, okay. Usually that peptidoglycan will be absent in eubacteria and it will be present in case of eubacteria, okay. So this is very, very important. So question can be expected with respect to the cell wall in archaea and eubacteria. So the plasma membrane lipids, so here it will be ether linked lipids, and in eubacteria it will be ester linked lipids. Anyhow, the plasma membrane is the higher proportion is lipid, but here it will be ether lipid in archaebacteria, and in the eubacteria it will be ester linked. Now, an antibiotics, so archaebacteria, so they are not sensitive to any of the antibiotics, okay. But here, these are all the bacteria, they are sensitive to antibiotics. So this is another important feature. And see here, the genetic machinery will be similar to the eubacteria, that is eukaryotes, that is RNA polymerase, they are having a transcription factors. This eubacteria, they are all the typical prokaryotic. Okay, so this eubacteria will be the prokaryotic and this archaebacteria will be similar to the eukaryotic. Another important thing will be the ribosomes. So the ribosomes will be 70S in both archaea and eubacteria. And this is the most important thing, the pathogenicity. The archaebacteria are non-pathogenic and this eubacteria are pathogenic. And the another important area where we can expect a question will be this examples: methanogens, halophiles, thermoacidophils. Methanogens means methane rich area. So those bacteria which can live in the methane rich area, we can call them as a methanogens. Halophiles means salt loving bacteria. Thermoacidophiles means where we can say higher temperature. Okay. Eubacteria means the examples in eubacteria is eukaryotes. Okay. So these are all the non-pathogenic bacteria. So the examples of the eubacteria will be E. coli, Bacillus and Streptococcus. Okay. So the evolutionary status it will be, it is considered as more ancient and this eubacteria will be evolved later than the archaebacteria. Okay. So this is the most important differences where we can expect the question, the difference between archaebacteria and the eubacteria, right? Now, another famous topic will be the difference between the gram-positive bacteria and the gram-negative bacteria. Okay, see. So now another important question will be the difference between the gram-positive and the gram-negative bacteria. Okay, so what is mean by gram-positive and what is mean by gram-negative bacteria? Okay, so these will be the classification. It is coming under the eubacteria. Okay, so whenever we are staining the bacteria, whenever we are staining the bacteria using the crystal violet. Okay, so they retain, these are all the bacteria, they are retaining the crystal violet stain and appear purple or violet. Okay, so those are all the bacteria, they are absorbing the crystal violet and they are showing the purple or violet color. So these bacteria, they are not retaining the crystal violet. So as they are not absorbing the crystal violet in their cell wall, they are appearing in a pink or the red color. Okay, so this is the major difference where we can see by seeing the color under the microscope, we can say that as a gram-positive bacteria or the gram-negative bacteria. Okay, so now the peptidoglycan layer. So the peptidoglycan layer, here it will be very thick and here it will be very thin. Okay, so as there will be a thick peptidoglycan, this layer is absorbing, this is the one responsible for retaining the crystal violet, and here as it is very thin, they are not, they, as the peptidoglycan layer is very thin, they can't retaining the crystal violet, so that it will be appearing pink or in the red color. Okay, so the outer membrane here in the gram-positive bacteria, the outer membrane will be absent. Here it will be present. And teichoic acids will be present in case of gram-positive bacteria and it will be absent in the gram-negative bacteria. Okay, and the lipopolysaccharides, so it will be absent in the gram-positive and it will be present in the gram-negative bacteria. This is considered as a endotoxin layer. Now, periplasmic space. So the periplasmic space, the space will be higher in the negative bacteria. So that is what a well developed. Here the space will be absent or it will be very narrow space. Okay. So with respect to the sensitivity to the antibiotics, whenever we are studying about this archaebacteria and the eubacteria, the antibiotics the eubacteria are sensitive to antibiotics. The archaebacteria will not be sensitive to the antibiotics. But in case of gram-positive and the negative bacteria, here the positive bacteria are sensitive to penicillin and the lysozymes. Okay, so as a gram-positive one minute, as the current power problem. See, the periplasmic space and the sensitivity to the antibiotics, they were sensitive to the penicillin and they are not sensitive, here the gram-negative bacteria is less sensitive to the penicillin. Not only to the penicillin, the gram-positive bacteria is more sensitive to the penicillin as well as to the lysozyme. Okay, and the toxicity. So here it will be mainly exotoxins and here it will be mainly endotoxins. The gram-positive bacteria will be the exotoxins and the gram-negative bacteria will be the endotoxins. And the flagellar structure. The flagellar structure here it will be there will be a basal body. So in the from the basal body the flagella will be arise. Okay. So from the basal body the flagellar structure will arise with 2 rings. But here the basal body with the 4 rings. So this is the major difference. And what are all the bacteria is considered as a gram positive and what are all the bacteria is considered as a gram negative. So the gram positive bacteria Staphylococcus, Streptococcus and the Bacillus. And here it will be Escherichia coli, Salmonella and the Vibrio. So these are all the bacteria. We can call them as a gram negative and gram positive bacteria. Okay. Now comes the cell wall composition. Okay, so the bacteria, their cell wall is made up of a peptidoglycan. In archaebacteria there will be no peptidoglycan. Plant cell is made up of cellulase, fungi cell is made up of chitin, and the animal cell, the cell wall is absent. So this is very important in an exam point of view. And with respect to the ribosomes, so 70 years and the eukaryotes will be 80 years, and the transcription and translation are coupled reaction in case of prokaryotes and it will be separate in case of eukaryotes. mRNA processing will be absent, which means post-transcriptional modification will be absent in prokaryotes but it will be present in eukaryotes. Okay, so these are all another important thing. And so in the prokaryotes, the DNA location, as I said before, then it is called as a nucleoid and it will be nucleus. And the introns are absent. Here introns will be present. So that is why post-transcriptional modification is happening. All these things will be discussed detailedly whenever we are teaching the molecular biology. Okay. So operons are present in prokaryotes and it will be absent in eukaryotes. Again it will be discussed detailed in that chapter. Okay. So but you should know operons will be present only in the prokaryotes and it will be absent in the eukaryotes. Okay. So another major difference will be, between a major thing will be in an exam point of view will be this mycoplasma and cyanobacteria. So this mycoplasma is the smallest living cell and there will be no cell wall and it is pleomorphic. Their morphology will be different when compared to the normal bacteria and the virus and it is resistant to penicillin. Okay, so these are all the most important characteristic features. And what is meant by cyanobacteria? Again it will be a prokaryotic. It has the capacity to do the photosynthesis function but there will be no chloroplast will be present in the cyanobacteria. Okay, so these are all one-liners, so very important in an exam point of view. Prokaryotes lack membrane-bound organelles, we know that, and the nucleoid, it is not equal to nucleus. You should not think nucleoid is same as a nucleus. Okay, so the region where the genetic material is present in prokaryotes, we are calling them as a nucleoid. Okay, so but there will be a proper nucleus, will the proper nuclear membrane in eukaryotes, so it is not same. Okay. So 70 years not equal to it is smaller than the 80 years. We should not think 70 years is smaller than the 80 years. It is not a right statement. Mycoplasma is the smallest living cell when compared to virus itself. So cyanobacteria is a photosynthetic prokaryotes. All these are the important thing. Now comes the MCQ. Okay, take 1 minute of time or pause the video and answer for this question. The cell wall of archaebacteria differs from that of eubacteria because it is absent, it contains cellulose, it lacks peptidoglycan, it contains a chitin. Okay, how the cell wall of archaebacteria differs from the eubacteria, we have studied about the peptidoglycan and the answer is option C. Okay, I have explained the thing also. Which of the following organism is correctly matched with its group? E. coli is an archaebacteria. Methanogens are eubacteria. Halophiles are archaebacteria. Cyanobacteria are archaebacteria. Okay, E. coli is an eubacteria. Methanogens are archaebacteria. Halophiles or archaebacteria is a right match and cyanobacteria is not an archaebacteria. So the proper answer will be option C. Now, third question will be archaebacteria are able to survive in an extreme condition. Why it has the capacity to survive in an extreme condition? It is mainly because what is the unique characteristic features? Because of the they are having an ether-linked membrane loops, making them extremophiles. So we have studied archaebacteria are ether-linked and eubacteria will be an ester-linked. So unique membrane lipid. So this is the important answer, important statement. Which of the following statement is true for the eubacteria? So eubacteria in the sense cell walls lacks peptidoglycan. Okay, generally non-pathogenic, sensitivity to antibiotics, live only in the extreme habitats. Eubacteria are sensitive to the antibiotics is a right statement. Live only in the extreme habitats. We can call them as an archaebacteria. Non-pathogenic, lack peptidoglycan. Again, it is a characteristic features of archaebacteria. Okay, so the correct answer will be sensitivity to the antibiotics. Methanogens are important because they cause disease. So this is coming under the archaebacteria. It is not going to cause any of the disease as it is not pathogenic. Fixed atmospheric nitrogen, again it will be this is somewhat related to the nitrogen cycle, nitrogen bacteria, fixing bacteria, and produce a methane gas. Of course it is right. Perform photosynthesis is not right statement. This option C is a right statement. Both archaebacteria and eubacteria share which of the following features? Membrane bound organelles, true nucleus, histone proteins, and 70S ribosomes. The correct answer will be 70S ribosomes. Okay. Now, which features makes the archaebacteria more similar to the eukaryotes? Okay, so this is a mode of nutrition, genetic transcription machinery, cell shape, and the ribosomes size. Okay, so which feature makes the archaebacteria more similar to the eukaryotes? So what will be the important characteristic features? That will be archaebacterial RNA polymerase resembles that of the eukaryote. So the correct answer will be the genetic transcription machinery. Okay. Now, which group is mostly pathogenic? The most pathogenic group of bacteria will be, whether it will be archaebacteria, cyano, and eubacteria, and mycoplasma. So which of the bacteria is most pathogenic means that will be the eubacteria will be most pathogenic. So archaebacteria will be the non-pathogenic in nature. Now next question will be antibiotics like penicillin act by inhibiting. So we have studied many of the bacteria are resistant to penicillin and many of the bacteria are sensitive to the penicillin. But if it is sensitive to penicillin, how the penicillin will works? Okay, by inhibiting protein synthesis, DNA replication, peptidoglycan synthesis, RNA transcription. So the correct answer will be penicillin targets the peptidoglycan which is absent in the archaebacteria. Okay, so penicillin act by inhibiting the peptidoglycan synthesis. So that is why the archaebacteria are not sensitivity to the antibiotics as it lacks the peptidoglycan. Now, which one of the following is an example for the eubacteria? So, eubacteria example, so methanobacterium, halobacterium, thermoacidophile and bacillus. So, all these bacteria, that is methanobacterium, halo and thermoacidophile, all these will be the bacteria which live in the extreme condition. So, it is coming under the archaebacteria. But the question demands the example for the eubacteria. So, the correct answer will be option D, that is the bacillus. Okay. So, thank you so much. We'll meet you in the next class.